Now let's study more about each class of immunoglobulin, especially IgG, which is one of the most important immunoglobulin because this forms the major serum immunoglobulin, about 75% in humans, and this is the most common type of antibody found in blood and also in extracellular fluid. It is almost distributed equally in blood and extracellular fluid. This is the only type of immunoglobulin that is capable of crossing placenta, which means this is the only immunoglobulin that passes from mother to the fetus across the placenta, which is the protective covering of the fetus. This is the only immunoglobulin that is capable of crossing this membrane, and hence this is one of the most important immunoglobulins, especially for the fetus, because the fetus itself do not produce any significant amount of IgG. These are secreted by the plasma B cells. The structure of the immunoglobulin molecule, which we just studied, is also the structure of IgG, since IgG exists as a monomer. If the diagram of an IgG is asked to be drawn, then the diagram that you have to draw is this diagram which we just studied, that is of the Y-shaped immunoglobulin molecule. It has a valency of 2, which means it is capable of binding to 2 antigens. Also, IgG contains less of carbohydrate than the other immunoglobulins, and it has a half-life of approximately 23 days, but we did not study this in the table because this 23 days is not fixed. The concentration of IgG and IgA, or the catabolism of these two, varies depending upon the pathogenic condition of a body or the disease condition of a body. Okay, coming to the functions, these are the major components of humoral immunity or the antibody mediated immunity. There are two types of immunity: the cell mediated immunity and the antibody mediated immunity, and these IgG form a major component of this antibody mediated immunity. They participate in most of the immunological reactions such as complement fixation, precipitation and neutralization of toxins and viruses. So they may be considered as a general purpose antibody which are protective against those infectious agents which are active in the blood and tissues both. And I forgot to tell that IgG not only exists as monomers but could rarely exist as polymer as well, that is multiple molecules together. Okay, and when they find an antigen of its specificity, they bind to it and form an antigen antibody complex which is capable of eliciting a response and prevent the antigen from causing damage through various mechanisms. First is by inactivation of antigen by forming an antigen antibody complex. Thus it prevents the antigen from binding onto the receptors of the cells that it intends to affect. Thus it blocks the antigen. Second one is by binding onto the antigen and labeling it to be acted upon by the WBCs, that is the white blood cells. Then the third mechanism is by aggregation of multiple antigen antibody complexes to form an insoluble complex and thus inactivate the antigen. Many such antigen antibody complexes come together and form an aggregation so that it can be later on phagocytosed by a macrophage. Okay, these are the three mechanisms by which IgG acts upon an antigen. Also, there are 4 subclasses of IgGs, that is IgG1, IgG2, IgG3 and IgG4, which are present in different ratios. That is, IgG1 is present in the highest percentage, that is it forms about 60 to 70%, of the IgG, whereas the IgG2 forms about 14 to 20% of IgG. IgG3 forms about 4 to 8%, and IgG4 forms about 2 to 6%. Second, the IgM. This is the third most common serum immunoglobulin, and it is the first to be produced by B cells when it is stimulated by an antigen. The first immunoglobulin that this B cell produces at the time of infestation by an antigen is this IgM. Structure: it is a pentamer, by which I mean that it has 5 such immunoglobulin units. The Y-shaped units, this IgM has 5 such units, and thus its valency is 10. Properties and functions: they are good agglutinating agents, and they help in clumping of antigens and eventual elimination. The third mechanism, which I spoke about in the previous slide, these IgM are very good at agglutinating. So the most of their immune function is through the third mechanism. The immunoglobulins bind to the B cell surface, and these act as the BCR, that is the B cell receptor, and these receptors are almost all the times IgM. These IgM perform this function of being the BCR. Then comes IgA. These IgA are present both in serum and in secretions, and while in serum they are present as monomers, whereas in secretion they are present as dimers, that is, two Ig molecules are present together, and hence you can guess what its valency is. In secretions they have another protein associated with it, which is called as a T piece or secretory piece. The IgA part of the immunoglobulin IgA, which is present in secretions, is produced in the plasma, whereas the T piece is produced in epithelial cells. Okay, and then it is added to the AGA as it passes into the secretions. The tears contain IgA. The IgA in tears are produced by the plasma, whereas the T piece is produced by the epithelial cells that are present in the eyes just before the IgA enters into the tears. It is the second most common serum immunoglobulin. The first common the immunoglobulin in serum was IgG. The second common is IgA, and the third common is IgM. In serum, it is second most common, whereas in secretion, this is the major class. As I said, the IgA are present in tears, saliva, colostrum, mucus, and hence are responsible for the mucosal immunity. Mucosal immunity, I mean to say the soft skin part of our body, which is the the lining of our oral cavity, the lining of our eyes, the lining of our nose, the inner lining. All these softer skin part of our body are protected because of these IgA that are present in the secretions, and they do not fix complement unless aggregated. They do not have the ability to of complement fixation unless they are in aggregations, and they can help in binding to some cells like polymorphonuclear leukocytes and some lymphocytes as well. Next comes IgD. They also exist only as monomer, just like the IgG. They are present in serum in very low levels, and they are primarily found in B cells, found on B cell surfaces, that is as BCR, and they do not bind complement. Their role is uncertain. Next comes IgE. Also exists as a monomer, and they are least common in serum. Like we studied, they form only about 0.004% of the serum immunoglobulins, and these are involved in allergic reactions like asthma, eczema, and other things. And their levels also rise in parasitic helminth diseases. This is nothing but the worm infestation. These are found in rise levels during such conditions, and they do not fix complements. Okay friends, with this we complete the topic immunoglobulins. Thanks for watching. Don't forget to like, share, and please do subscribe so that you are updated when I come up with the next video. And feel free to post your comments, queries, or suggestions in the comment section below. Thank you.